Okay, good afternoon. I hope everybody can hear me quite clearly. My name is Tim Corson. We are calling in from Vienna today, and we have another edition of the Early Career Members Network webinar series, which we are part of the Metabolomics Society. This is the 12th session, and today we've got a talk on quantitative metabolomics. If you're not too familiar with the EMN and what we do, we're part of the Metabolomics Society. If you want to know a bit more about what we are doing, you can connect with us online through a number of different platforms via Facebook, Twitter, LinkedIn, or you can email us directly. We're there primarily for young members, and this webinar series is primarily designed to engage with people and get some educational ideas out there for metabolomics. Speaking of, if you really want to get involved and meet new people, and many of those people have been working in metabolomics for a few years, or they're also young members, it's a nice idea to get involved in the Metabolomics Forum. And here you can see there's a lot of interesting topics and discussions to be had there. So I would encourage you to join if you haven't done so already. Later on this year, you might also be aware that the Metabolomics Society is having its annual conference in the beautiful climes of sunny Brisbane in Australia. You can see here that there's still time to submit abstracts. And if you manage to get down there in June or July, although it's winter time in Brisbane, it should be nice and sunny. And there'll also be a lot of good scientific content as well. The EMN is actually promoting 4 workshop topics this year. So these are particularly designed towards engagement with young members. As you can see, there's a topic on career development. We're also discussing experimental design in metabolomics, data processing, and finally some statistical considerations and pathway analysis strategies. So there's some extra content there which we are trying to be involved with to engage directly with young members. Today I have the pleasure to introduce Professor Stephan Hann from the University of Natural Resources and Life Sciences in Vienna. Stephan has been working in metabolomics for a number of years now, and particularly in the area of quantification. And it's my great pleasure to introduce him, and he'll take over now and be presenting his topic.
Hello everybody, I hope you can hear me. My name is Stephan Hann, and I want to thank Tim for his kind introduction. As you have seen, the title is about quantification today, about metrology, measurement uncertainty, and I will try to give you an overview of modern concepts and the state-of-the-art concepts for validation of analytical methods, and of course validation is not only a topic in metabolomics, it's a topic in analytical chemistry, which means that it is applied and has to be done in many different fields. First of all, I want to thank my co-authors, contributing authors here who have contributed with their work. Teresa Meiringer, she has done a lot of measurements and uncertainty calculations, and is currently calculating quite a huge project on measurement uncertainty. You will see a glance of it later on. Christina, who is doing our GC development and applications, and Professor Wegscheider, who is a past chair of Eurochem. I will come to Eurochem, to this institution later on. And I'm happy that he helped me to set up this talk and gave me support. So let me immediately come to some important documents, key documents for metrology validation, uncertainty, and accreditation. There is 3 documents, or even 4 documents I want to show you here shortly. I put the links below, so these are downloadable the WIM, International Vocabulary of Metrology, is a very important document, which is the base of most of the validation and uncertainty documentation, which is defining all the terms necessary for these procedures. Then we have the guide, quite new release of the guide, the fitness for purpose of analytical methods. Unfortunately, there are not too many documents around or available which give you a clear procedure and a clear workflow for validation of analytical methods. So this is the one you should follow if you want to do proper validation of your methods. And next, and this is something which is also part of validation, quantifying uncertainty in analytical method. This is quite a complex topic, but I think the guide I'm showing here is giving a really good support, or supports you really good to calculate and quantify, estimate uncertainty in the analytical measurement. And I will show you at the end, or was at the end of my talk, how we apply this guide. All three of these guides are the base of the ISO 17025. The ISO 17025 gives general requirements for the competence of testing and calibration laboratories. There is a new version coming up this year, and this is the so-called accreditation standard, which means it's defining all the requirements you need if you want to run an accredited lab. I know that most of you are working in research, in science, so accreditation is possibly not a topic in your lab, but of course all the labs which are doing pollution control, food control, and so on, doping controls, are following this guide, these standards, and are accredited, of course. So another source I want to mention, an additional source, is TrainMIC. TrainMIC was a program, a network within the European community. The documentation of TrainMIC still exists. It was a training network which was aiming at harmonization of metrological procedures and metrology in general in the European Union. So you will find, if you follow this link and this publication, you will still find open source content from TrainMIC, which is for sure very valuable. And you will also see, as soon as the TrainMIC logo pops up in the right corner, you know that this content is from this TrainMIC documentation. Okay, so let's start. So I will give a, let's say, general definition, general introduction on validation terminology now, and show you some sources and further reading possibilities. And then we will go on with some specific examples. In principle, the 17025 has three important topics, covers three topics, which is validation, traceability, and uncertainty of results. I will not go into detail at all the slides here. I show you, so that's for further reading, and I will just add some important comments if necessary. Validation is the validation process depends on what you want to validate, or what you have to validate, respectively. It depends on if you have ISO or DIN or EN standards available. So if you do have such standards, if you can follow such standards, you do not need full validation. You just do verification. You show that your method is fit for purpose, and that you can follow these standards and achieve all the characteristics, the performance characteristics. A very important topic is traceability. I think everybody knows what SI units are, and of course the Bureau International des Poids et Mesures is responsible for traceability and for setting the SI units. I will come to that later on. And the third, as I said at the beginning, the third milestone in the 17025 is uncertainty estimation. The certainty 17025 demands for uncertainty estimation, and each lab which wants to get accreditation needs to show that procedures for quantifying uncertainty are available for all the parameters a lab is determining. The definition of validation finally means or says that you have to show that your method is fulfills the requirements for an intended use. So of course you have some requirements. If you run a research lab, if you do a research study, you will know into which concentration ranges your metabolites will be present, at which concentration ranges your metabolites will be present. You will know which matrices you have to analyze, and so on and so on. So you have some requirements, and these requirements you have to prove that you can fulfill these requirements with your method if you want to validate your method. What does it include? Of course, you first of all need to know about your analytical requirements, the intended use of your procedure. You determine the procedure characteristics. We will come to this specific procedure characteristics later. And then you will check that the requirements can be fulfilled by the procedure, and you will state that your procedure is fit for the intended use. Then your validation process is finished, and you will deliver a validation report. What's the aim of validation? The aim of validation is that it's a process of establishing performance characteristics. You should find out the limitation of a measurement procedure, and you should identify influences which may also influence or have an impact on the final result of your measurements. So this is the term which will show up later, is ruggedness, which means how rugged, how robust is a procedure in terms of influences. Finally, you want to state in your report which analyte can I determine in which matrices, in which measurand. Measurand is an expression which is used in this documentation quite often. The measurand is the analyte including the matrix. So you're analyzing amino acids in cell culture supernatants, for example. So that's a measurand, analyte and matrix. And the question is also which interferences do I have, and what is the selectivity of my method with regard to these interferences. Third part is with the conditions I've defined, what uncertainty can be achieved. What is the uncertainty of my value? I will come up with examples later on. Again, it's the process of verifying that the procedure is fit for purpose. Okay, so what is the role of this validation? Why do we need this validation? Of course, you are performing measurements independent if you are in a food control lab or in an environmental lab or in a medical lab or in an industrial lab, or if you are working in science. You will make decisions because or in connection to your results. So the reliability of your results will finally determine your yeah, let's say your success, and of course you need reliable results in order to assess, for example, the compliance with laws and regulations. So the customer who is demanding validation can be the government, for example. It can be industry. It can be a research lab. It can be a medical lab. Finally, you need or you want to enable decisions, and you want to come up with reliable data and reliable results. And you can only prove that your results are reliable when you are using a validated method and when you have validated your method. The guide, as I already mentioned here, we are using for method validations is this 2014 guide, Fitness for Purpose of Analytical Methods. You can download it under this link. Okay. If you, as I said at the beginning, you do not need full validation, base validation, if you have available standards. Of course, this is not the case in metabolomics, unfortunately. And this is really a difficult situation, because if you want to, as we know, if you want to generate reliable data, you need to validate your entire procedure. If you would have standards available like DIN, ISO standards, CEN standards, this would mean that you just follow these standards, follow these procedures, and you have just to prove that you can fulfill the requirements of these procedures. There are also, let's say, a little bit weaker procedures published. These are not standards, but they are published by reputable organizations like EPA or so, or ICH. You can follow these standards as well, but you need a higher amount of validation effort if you follow such procedures. What is not accepted if you have an accredited lab and you want to get accreditation for a certain procedure which has been described in scientific literature, this will not be accepted as a standard procedure or as a standard method, which means that you have to perform full validation on a scientific procedure which has been published by your own lab or by somebody else. Okay, short note, traceability or short example. I think everybody is aware of SI unit, the Système International d'Unité, and BIPM is heading all and organizing all this. Just as an example, the unit of mass is traceable to the kilogram, which is situated in Sèvres, close to Paris, and this is the SI unit for the mass. So be aware that if you want to demonstrate and you want to prove traceability, that the balances you are using for weighing your samples have to be traceable to this kilogram. So this means that you need calibration documentation, calibrated weights, and so on and so on. Only then you are traceable to the kilograms. Same regarding volumetric measurements. You need also to be traceable with volumetric measurements. You can do this via the balance, which means that there is an ISO standard which allows you to calibrate or to check your pipettes. This is traceability, and you need to prove traceability. And of course, everything I'm telling you here is not only necessary when you want to go for an accreditation or want to become an accredited lab. It should also be done in any lab which is measuring analytes, quantifying analytes, and is producing data. Okay, what has to be validated? I said already, I was mentioning full validation. Full validation means that you have to validate the entire procedure. You have to validate the performance characteristics from sampling, if you perform sampling, over sample treatment, instrument and calibration, and measurement, data processing, as well as data transfer and reporting. So the last two steps are also important. You also have to document that and to prove that you don't make any mistakes while processing your data, or that you are correctly transferring the data into the final report, which is in a scientific lab, the publication. So there needs to be trackability proven for the data coming out of the measurement system, of the LCMS system, GCMS system, into the scientific publication or into the analysis report. Of course, also the concentration range you are working in needs to be validated, and all types of matrices. If you are able to analyze cellular extract from ethanolic extraction procedures, this does not mean that you are able to analyze or that you are allowed to analyze cell culture supernatants with the same procedure, same sample prep procedure, and so on. So the matrix you are analyzing needs validation as well. So validate all relevant types of matrices you are analyzing in your lab. You have to perform validation when you develop a new method, as I said, when no standard method is available from ISO or similar. If you change your method regarding matrix, regarding range, regarding instrumentation, you have to do revalidation of certain parts of it at least. If you notice that the daily results, the daily controls and so on do not comply anymore, of course you also have to think about, of course, first of all you will check everything and try to find the source of the systematic error or so. But finally, you will also need to re-validate parts of the method. And as soon as you change the environment, which means as you move an instrument from one lab to the other, you will need to do or to at least verify the validation you have performed for this certain procedure and instrumentation. Now we come to a little bit closer to the procedure itself. What are the classical validation parameters? Many of you will know them. Of course, the working range, sensitivity, selectivity, limits of detection and ruggedness. These are performance parameters of the procedure which have to be assessed. And then you have the property of the result, which is on the one hand traceability, precision, trueness, and uncertainty. So these are the parameters I will explain to you within the remaining minutes we have. The example we took is targeted targeted quantitative analysis for metabolic profiling of microbial cell factories. We are analyzing lots of samples coming from such experiments. We are analyzing CHO cells and cell cultures. We are analyzing Pichia, so yeast cells, and so on, coming from biotechnological research. So we here at our university, we have a strong biotechnological department which is mainly aiming at the optimization of microbial cell factories, which are used for production of a wide range of chemicals and pharmaceuticals, also known as wide biotechnology. So usually this is our sample here, and what we get out of such a cultivation is different types of matrices. Of course, these matrices are all challenging. We get intracellular samples, we get extracellular samples, whole broth samples, quenching solutions, washing solutions, and so on and so on. So this means that, yeah, as I said, it's quite a challenge, and we have to validate for all of those. This is a workflow for profiling of intracellular metabolites of the central carbon metabolism. We are using on a routine base in cooperation with biotechnologists and also industry, biotechnological industry. And as you see, we are mainly using LC-MS-MS and GC-MS-MS, so we use triple quad equipment, tandem MS equipment in a targeted mode. Two different liquid base separations, helic and reverse phase, and derivatization. I will show some details on this method later on. Then we perform data processing, deep integration, and report. There is one special thing I want to mention here already. We are using labeled internal standards. I will come to that later, and we are adding this labeled internal standard as early as possible. So here you see, so this is the conventional cultivation here, our real sample, but at the same time we are also, from time to time, if we run out of this standard, we are producing uniformly C13 labeled extract of our organism, which is yeast in this case, and this extract contains all the metabolites, so the entire biomass is fully labeled, and that's a perfect internal standard for our application. I will come to this in a minute. After the or during the cultivation, if you want to do time-dependent sampling, we are quenching in cold methanol. So this is the competence of our partners, of the biotechnologists. They have to do sub-second sampling in order to quench the metabolism. And after that, we have rapid filtration of the cells, which is not so crucial during internal standard production, because the internal standard is not quantified. It's just our reference, as I will explain in a minute. But here, of course, it's important that the metabolite concentrations, that metabolism is stopped, and that the metabolite concentrations are already stable, and that we don't have degradation of compounds and so on. Afterwards, we are performing boiling ethanol extraction or cold extraction techniques in the case of CHO cells. And during this extraction step, we are already adding this uniformly labeled internal standard. As I said, I will come to that later. This is the GC-MS system we are using. We are doing derivatization, of course, for primary metabolite. We are derivatizing with ethoxymation and 3-methylsilylation, quite similar to the FIN protocol, but we are using ethox instead of methoxymation because we get a better separation of the phosphorylated compounds there, phosphosugars. As you can see here on this slide. Of course, the final aim of the entire procedure is we want to obtain accurate concentrations. And you can perform several different quantification strategies to derive concentrations and to measure concentrations of metabolites in these samples. And I will go through them to 4 examples now. One is let's say the simplest approach is external calibration. I think everybody knows how this works. I don't have to go into detail there. I just want to point out that the disadvantage, of course, is if you have a heavy matrix and you're calibrating in water, this will not fit together. So you will have suppression, you will have signal suppression, you will have many problems with your matrix, and of course an ideal standard used for calibration does not represent the same matrix and does not result in the same suppression. So this means that this is prone to error. What you could do is you could perform so-called matrix matching, but it's not easy to find a matrix free of your analytes, especially in bioanalysis. So a better approach is standard additions. If you go for so-called internal calibration. With internal calibration, you are adding your analytes of interest at different concentrations to the sample. I think everybody knows about that as well. Of course, you have some drawbacks here as well. First of all, you are multiplying your analysis time because you have to analyze your sample several times, and so this means you will apply this procedure only if you have a low number of samples, so very small campaigns, or for validation purpose. So if you want to have an intercomparison with an independent quantification method. As already mentioned, internal standardization is the better way to go in our opinion. There are two possibilities for internal standardization. So that's external calibration with internal standardization. You try to compensate for matrix compression, matrix enhancement, and other effects by adding an internal standard. The drawback is, in this case, that your internal standard, of course, does not exactly represent your analytes. It's eluting at a different time, and it will also have a different degree of suppression and enhancement effects because it is not the same compound. The better way to go here is standardization with labeled internal standards. These are chemically identical, almost identical, and you can use them for normalization. So that's the high-end method. In elemental analysis, it's known as isotope dilution. There it's even simpler because you know the concentration of the internal standard you're spiking into your sample, and you just over the ratio you can determine the concentration of your analyte. So this is the spike blend ratio, or the blend ratio in your isotope dilution sample. If you're interested in isotope dilution in the concept, just go into literature search. You will find a lot about that. Isotope dilution is recognized as a primary method of measurement because it's a ratio measurement. And ratio measurements in this case we see here are of course beneficial because you are measuring quasi-simultaneous with your triple quad, your analyte and your internal standard, and you're just going for a ratio, and this ratio is also on the y-axis of your calibration graph. How can you, well, where can you purchase the internal standards? Or most of the companies have them available, synthetic compounds. You can also buy already synthetic mixtures of compounds, and what is getting more and more modern is the production of uniformly labeled cell cultures, and these extracts are also available with the advantage, as I already said, that if you have a uniformly labeled cell culture, you will have all your metabolites, the ones you're analyzing, but also the others which you're not analyzing or you will analyze in the future. You will have them available as fully labeled compounds, and that's highly beneficial for the reliability of your measurements. Okay, we come back now to the validation list, and I will go through some, let's say, more common terms and vocabulary here. Working range here, I just want to point out that the working range has an upper limit and a lower limit. And the upper limit of the lower limit of the working range is the highest standard you are using for calibration. You do not know about saturation effects, nonlinearity after this point here. So you are not allowed to measure outside of the upper limit of the working range. The lower limit of the working range is not the lowest calibration standard, it's the limit of quantification. I will come to the limit of quantification later. So the limit of quantification is the limit. From this limit on, you are allowed, so or above this limit, you are allowed to give a quantitative value. If you're reporting values between the limit of quantification and limit of detection, of course you have to state that this is in this range here. How can you calculate the limit of detection, limit of quantification? You will find about 100 different procedures in the internet. Of course, this is a topic which is discussed all the time. The EuroChem guide now, we are very happy about this. The EuroChem guide, the 2014 edition, gives a clear procedure for assessment and for calculation of limit of detection, limit of quantification. As I said, it's free. You can download it, and I would recommend to follow these procedures. Basically, you are determining the standard deviation of repetitive measurements of a blank. Yeah, so you are calculating the standard deviation, and then you just calculate this standard deviation by the square root of replicate observations you are using for your results. So that's important. It's not the square root of the replicates you are using for calculating the standard deviation. Be aware of that. In case you are using blank subtraction, it gets a little bit more complicated. You have to also insert the number of blank observations you are using in your procedure. This is also always related to the procedure you are using. After you have calculated this index S0, you multiply it with 3 for the limit of detection and with 10 for the limit of quantification. So, some comments regarding this procedure. You need to measure procedural blanks. Of course, if you have a procedure, sample preparation procedure, sample enrichment procedure, the blanks have to go through these procedures as well, or procedural steps. Second, if you do not have real integrable peaks in your blank, in your chromatographic blank available, you have to prepare pseudo-blanks. So you have to spike near zero concentration in order to get a signal, an integrable signal. Be sure that you take the right number for NB. That's the number of blanks averaged in routine use. It's not the number of blanks you have used to determine the standard deviation. If you are going for ratios, as I've shown before, if you are doing a normalization to an internal standard, of course the blank signal has also to be normalized to this internal standard, and then you calculate S0 out of these ratios. And finally, and this is maybe the hottest topic, many people are using the noise in the chromatographic baseline, and they're doing signal-to-noise calculations, and so you cannot use this for calculation of a standard deviation of the blank because it does not include the peak integration. So you need to integrate a peak, and from this peak you can calculate the standard deviation from this area, because everybody is going for assessment or calibration via area, and this means you need, and the integration process needs to be included in order that you get representative standard deviation. So you are not allowed, following this guide, you are not allowed to calculate your standard deviation of the noise from the noise region of a chromatographic baseline. You need a peak signal. Okay. One term which is also popping up very often is robustness, which should be called ruggedness. Ruggedness is telling you how stable your process is if surrounding parameters are fluctuating. For example, pH. How important is the pH of your sample? If the pH of your sample changes, will this have an effect on your results? Temperature. Can you run your method in your lab although it's not thermostated? So if you don't have air conditioning in your lab, you will have fluctuations of temperature. If you want to run a validated method there, you have to prove that the results are not influenced by this temperature fluctuation. Then you have proven ruggedness related to temperature changes. So if you mention ruggedness, you always have to add for which parameter did I check or did I determine ruggedness. Going back to our list, we come now to the property of the result. There are also some long definitions you get in the WIM regarding precision. Precision is very often mixed up, or let's say the terms repeatability and reproducibility are very often mixed up. So be careful if you use these terms. There is a clear difference between repeatability and reproducibility. Mainly, repeatability means that you are doing everything under the same conditions. So you do not change anything. And you do it in a very short period of time. Yeah, so it's really a repeatable injection of your sample finally. Or you're running several sample preparations in parallel, and then you have repeatability. Reproducibility is something different, because with reproducibility, a condition changes. So this means you are doing it at different locations, which means different labs are delivering the data. You have different operators, you have different measurement systems, and so then we are talking about reproducibility. So be aware of that. Same with trueness and bias. There are also clear definitions for trueness and bias. Trueness is not a quantitative term, but bias is the quantitative term, and there is a clear definition. It is the distance or the difference between your measured value, here in blue, and the reference value. The reference value is the best estimate of the true value. I think you will all agree, nobody knows the true value, nobody knows the true concentration of your metabolite in your sample. So you have to find a reference value, and then you can determine how, if you have a bias, if your lab, your method, your procedure is biased regarding this reference value. Where can you get this reference value from? Well, let's say the best reference you can get is a certified reference material. As I said, these are produced and available at NIST, at LGC, and many other providers. So you should be able to find reference material. Also, I have to say, in metabolomics, this is not so common, and there is a lack of reference materials, and I think the community should work on that. We should try to set up programs which aim at the production of such reference materials in order to generate best estimates of the true value, and in order to give the labs the possibility to determine the trueness of their methods. Other possibility is proficiency testing, round robins. So you compare to other labs, or you make method intercomparison. So of course, that's a ranking. So the best you can get is a certified reference material or a reference material, which has not a clear certification, and so on. Of course, standard edition experiments can also be used to generate a reference value, but be aware that standard edition, if you go for intracellular metabolites, does not make sense because you are not aware of the efficiency of your extraction procedure. And of course, this will not be compensated by your standard edition. Finally, do not mix up trueness or precision with accuracy. Accuracy is something different. Also here, the WIM gives a clear definition. Closeness of agreement between a measured quantity value and the true quantity value of a measurement. And the note 1, there are several other notes, but this is the most important one in the WIM. It's not a quantity. Yeah? So it's not a quantity. You cannot give a number to accuracy, which means that it's just a description of your procedure. It's said to be more accurate when it offers a smaller measurement error. So that's the clear statement you will find in WIM. Okay, so this is a short example. I have to have a look and ask how much time we have still. 5 minutes only? Okay. I will be quick. 10 minutes, okay. We took an example from metabolic flux analysis, so I will not go into details here. There's a lot of literature and research done in this field. Basically, regarding metrology, it's the measurement of isotopolog ratios. So definition of an isotopolog, I think, is clear: molecule that differs in terms of their isotope composition. So it means it's not positional. With isotopomers, you have the same isotope composition. So this would be isotopomers here. They have 2 carbon-13 atoms here in the backbone, but at different positions. This is isotopomers. Yeah? And isotopologs have an increasing number here of C13 integrated. We are looking at the ratios of the chromatographic peaks we get with our flux methods, and we are then calculating isotopolog distributions. So that's quite a simple process, but we are of course interested about the trueness of these measurements. We can do this with GCTOF MS or with LCTOF on the different metabolites here. And here is our GCTOF method we are using, and this is the LC method. And here you see such an artificial pattern. I will come to that on the next slide here. You have the different metabolites. And we are interested now in how is the bias of our method here. How can we determine this? We need a reference value. And we found a very good publication of the group from Professor Portet in Toulouse, where they have produced a reference material by incorporating a 50-50 mixture of C12 and C13 into a yeast fermentation. And we have reproduced their paper and their method, and we have generated one of these materials where we have a distribution of the label into the different metabolites depending on the number of carbon atoms they carry in their backbone, following Pascal's triangle. So this means that if you have 2 carbon atoms, you will have this distribution. If you have 3 carbon atoms in the metabolite, you will get this theoretical distribution, and so on and so on. So we have now a best estimate of the true value here with this isotopolog fractions, and this is the result of our measurement. So the green bars are the best estimate of the true value. So this is our predicted isotope log fraction, and we can then determine the bias between our measurement and the best estimate of the true value. So that's high, and here you see the GC results for the same or similar analytes, and how we can then assess the trueness of our results. Last topic of this webinar is uncertainty of measurement. As I already said, uncertainty of measurement is quite a complex topic, and looking at the documentation we have available, the QAM, which I have cited here, Quantifying Uncertainty in Analytical Measurement, 3rd edition, is valid at the moment, gives you a clear description and definition, but it gives you also very nice examples where, let's say, the entire topic is explained in a practical way, and where you can follow the determination of measurement uncertainty. Why are we doing that? Why are we interested in uncertainty of measurement? Why don't we just take precision? Yeah? Why don't we just take the repeatability we get in our own lab and report this repeatability? Why is uncertainty something different than precision? Because it's bigger usually. So as you can see here, it's just, of course, an abstract example. When you want to do differential analysis, and that's what's done in non-targeted metabolomics very often, relative quantification and so on, and you want to see a difference between two groups, for sure you have to consider the uncertainty of your results. If you, of course, people like to use precision solely, but as you know, precision does not represent the entire uncertainty of your procedure, because the factors which are influencing precision, or the variability which is caused by these factors, is not representing all possible influences of uncertainty. So it may be that you end up with the same uncertainty, or with an uncertainty in the same range as the precision of your procedure, but usually the uncertainty is much higher than so looking at these arrow bars here, than the precision. How can we now estimate, and how does this work, that we find out uncertainty, and why are we doing that? Of course, not only in differential analysis, but also if you control limits, if you want to control limits for pollutants in food, or if you want to do doping control or contaminant control, whatever, you need to report your results with uncertainty. Precision alone is not enough. That's something which is clearly stated in the 17025. If you want to work in an accredited environment, you need to report uncertainty as well, because of course uncertainty, and not only your value alone, is determining if you have exceeded a limit value or not. So, and by law it said that the limit value is only exceeded if it is exceeded including the uncertainty. So you need a really reliable process to determine the uncertainty of your results. How does it work? As I said, in the GAM, you have a good explanation and very good guidance there. In principle, you need the equation which is delivering your final result. If you have this equation, a calibration function, including the volumetric or the dilutions you are applying, or if you're weighing in some sample, and so all these steps you should have in your formula, and this is the so-called model equation. Then you need to give uncertainties to these different input quantities, and then you can propagate the uncertainty. So this is the procedure which is and the process which is given in the quant, and you can follow this process. Of course, if you read it here, it's quite abstract, but you will understand and you will be able to follow this process when you're looking at the examples which are given in the appendix of the guide. I will not go into detail here. I will just show you something which we are using in order to find out which factors and which input quantities are contributing to our absolute fluxes we are calculating in metabolic flux analysis, C13-based metabolic flux analysis, from isotopologue log fractions, basically. So I want to draw your attention to this part here. So which input quantities and which sources are we considering here? Of course, we have a detection system, which means that we have counting statistics leading to variability of our measurement. We have in front of the counting statistics, we have the entire ionization and ion transmission process. We have, when we have counted our ions, and chromatogram is generated, and we are doing the peak integration on this chromatogram. So if you do if you are integrating the same peaks 10 times, you will see that also there you have an uncertainty, you have a standard deviation on the areas, and all these input quantities are then combined, and they lead finally to the uncertainty of absolute flux. Of course, it's much more complicated here. I will not go into more detail, but of course you have not only the measurement when you go from a metabolic flux analysis, you also have other sources of error, the entire cell culture and tracer study you are performing up front, and so on, are of course influencing the total combined uncertainty. So just for the measurement, as I've shown you before, if you remember the isotopologue log fractions, so here we have 5 ions we are measuring from one compound, 5 isotopologues. We are summing up all of the chromatographic signal and use them to divide one. Here we have selected the A4 isotopologue, which is divided by the sum of all the areas of the entire isotopologues we are measuring for this compound. That's the isotopologue fraction, and then we are giving errors to all these fractions, which are considering the different here ionization process, transmission process, reliability of automated peak integration, and so on. So we are assigning standard uncertainties to these different areas here, and then we propagate these errors, and what we're using here is Monte Carlo propagation. You have software available for Monte Carlo error propagation as Excel add-ins, and also on the NIST homepage you will find a tool to propagate via Monte Carlo, and this error propagation is then leading to an uncertainty here below. So the uncertainty of this isotopologue log fraction would be 1.5% relative standard deviation, and you will ask after all, okay, why is it good? What is the benefit of all that? Because I could also multiply my precision with 5, or the standard deviation I have on 5 replicates. I multiply it with 3 or 4 or 5 or whatever, and I estimate everything. Okay, you can do that if you are aware of the process, but the drawback is you will not find out what are the sources for the uncertainty. And if you are doing this budgeting, as I have shown you before, so if you have understood your process, and if you have understood which sources of error you have in your process, you can also quantify the contribution of these errors. And then you know where you can optimize your procedure. So where do I need to optimize my procedure, and how can I reduce the uncertainty of measurement? That's the entire goal of it. It's understanding the sources, it's defining the sources, and if you want to define the sources, you have to find out everything about the process. You have to understand your method, and then you can assign uncertainties to these different input quantities. And finally, if you propagate them, you can find out which parameter has the highest contribution to the uncertainty and how you can optimize your method. Okay. Finally, here are some references, just a selection you can use, but of course, this is more about application and let's say more metabolomics centered. Of course, I recommend, if you are interested in this topic and if you want to have clear guidelines for validation and for uncertainty calculation, please look at the documents I've shown you at the beginning. I want to say thank you to my group and to everybody who is contributing to our daily work and to the really nice atmosphere we have here. I want to thank all of you for your attention, and I think now we have time to do some question and answers, and I'm looking forward to your comments. Thank you very much.
So thanks, Stephan, for the great webinar. We have a few questions that have come in. A few of them are quite specific, and we've got one here that's about the use of vendor software to perform a lot of validation procedures like limit of detection and limit of quantification and linearity. And the question was, can you use these directly for validation, or do you need to check these things manually?
No. It's absolutely no problem to use these procedures. Sorry, I have a technical problem here. Okay. Can you hear me? Good. Of course you can use Excel sheets for the validation. If you look at the accreditation standard 17025, all Excel sheets you are using, of course, have to be trackable. So all the calculations you are doing and everything. So this is, of course, something you have to be aware of, that you have to validate your calculations, and the documentation has to be fit for the purpose. But it's that there's absolutely nothing to say against doing the calculations in Excel.
We've got another question here about GCMS-based quantification. The question is, it involves multiple steps, including two-step prioritization. So how can you assess the coefficient of variation, and how can you, in other words, the coefficient of variation or RSD cutoff should be different for the two different platforms if you compare to LCMS. So how do you take that into consideration?
Of course, you will have different variation coefficients. You will have different repeatability precisions for the two systems. It's absolutely no problem as long as you report them and as long as you determine them correctly.
Got another question here about triple mass spectrometry, which is if I run a blank and I don't get any signal for the noise, how am I going to be able to estimate the noise from my LOD?
That's what I try to explain on one of the slides. You have to fortify your samples. So you have to your blanks. So you have to generate something I would like to call a pseudo blank that you get an integrable signal. So you spike, I would say, estimated 1/10 of your estimated limit of quantification into your sample, and then you will get an integrable signal, and you can calculate standard deviations.
Okay, we might just have time for maybe one more question. I've got quite a few here, but I think we should not go too long this afternoon. I'll just have to pick one from the last four that have come in. Sure, I have one here. So here's the question: what's the most reliable value of relative standard deviation?
I don't see it. It depends on your procedure. You cannot answer this question. It depends really how complex your procedure is, what is your matrix, at which concentration levels are you working. So many different influences will determine the standard deviation. I cannot answer this.
Okay, we'll just do one last question. What are your suggestions for researchers performing global metabolomics who do not have access to isotope-labeled materials, particularly for the biofluids such as plasma and urine?
Do as many as possible. I mean, of course, it also happens to us from time to time that the signal in our labeled extract is not strong enough for a certain metabolite, because we are applying these labeled extracts also to clinical samples. So we also can use the yeast extract, the fully labeled yeast extract, for quantification of other matrices or the same analytes in other matrices. But if the signal is not strong enough, we are using, let's say, the closest, chemically closest compound for normalization. Finally, normalization is always the better choice than going for recovery, determination, and doing external calibrations without internal stabilization.
Okay, I think we'll probably have to call the day there. Thanks very much to Stephan for giving that webinar today. You'll be able to access and download this webinar online. That we'll be able to set that up in the next few days. Just want to thank you all again for your attendance and participation today. I'm sorry you couldn't get through all of your questions, but I'm sure if you send Stephan an email, we'll be able to answer those by email.
Okay, so I think you have my email on the screen, and I thank everyone for your attention, and have a good day, afternoon, evening, and a nice weekend.